Welcome to What's New in MPMI, the virtual seminar series of the MPMI Journal. As everybody is joining our virtual room, I hope you'll type into the chat where you're from. I know we have attendees from all over the world, and it's exciting to see where you're all from. So type into the chat. We have Sridhar Raghunathan from Chennai, India. Who else is out there? Ann Mathias from Chapel Hill, North Carolina, US. Brian Push from Elma, New York. Hi, Brian. And Jakub Wojsokinski from Lublin, Poland. Carolee Bull from State College, Pennsylvania. Pawan Kumar from University of Chicago. Yejin An from Seoul, South Korea. Amelia Reddy from Montreal. Canada, oops, I'm going to miss some. Alberto Valencia Botin from Jalisco, Mexico. Several people, Deepak from East Lansing, Michigan. Arizu Rahimi from Germany. Wakoi Kim from Seoul, South Korea. We have Bolzano, Italy. Krista Anju from USA. Noriko from Japan. Hi, Ki. Hi, Ki-Hun Sun from Seoul. And number of people, Miriam Valenzuela coming in from Valparaiso, Chile. It's 10 p.m. Welcome. So we have people coming from all around the world. It's always great fun to see who is here and who's joining us. For those of you who just joined, this is What's New in MPMI, the virtual seminar series of the MPMI Journal. I'm Jean Harris. I'm the host and organizer of this series, and it's been a great pleasure for me to be doing this for the last 3 years. It's kind of amazing. It's been going so long. The goal of this from the beginning was inclusion. It was an effort to bring our science from our journal to our very large international community that's across the world, and to bring it to everybody, make it free of charge, even as our journal then became open access. So this is science that we are doing that's for everyone. And you can also take a look now at the papers and read those for free as well, open access. So that's really one of the goals, accessibility. And also, we're an international community. Plants all over the world, microbes are all over the world, and our community is all over the world. So it's particularly international today because I am joining you from Vermont in the East Coast of the United States. Karen, our IS-MPMI staff member, is from California on the other side of the United States. And we have Hyelim Jeon joining us from Seoul National University in South Korea, and Cécile Segonzac, also from Seoul National University, who is joining us from her sabbatical in France. So right now, on one call, and that's without even any of the participants, we are wrapping around the globe. So seminar at all hours, morning, evening, afternoon, middle of the night. So anyway, that's just a little fun aside. So today's seminar is going to be a joint effort by the two authors of a very nice review on bacterial effectors and the endomembrane system. And this is part of our most recent focus issue, focusing on the role of the endomembrane system in plant microbe interactions. And it's, I think, a really important area for us to be thinking about. For so long, we focused on what was happening at the surface of the cell, what's happening right when the two organisms first interact, or what happens after effectors are secreted into the cytoplasm. And of course, there was this whole area we were missing, this whole secretory pathway that we now know is phenomenally important, and also the target of many effectors from the microbes. And this is, Cécile was pointing out, that the next focus issue for the MPMI journal is effectors at the interface of plant microbe interaction. So very timely, and I think you'll enjoy this. And this review that they wrote is already turning into a really valuable resource, because there's so much new work happening, and they've pulled it all together in one place. And really interesting. And by having that all paired together, their article takes them to a number of really important questions at the end. And before we move on to the talk, I want to remind you that we do have another talk coming up next month in December by Hasna Boubakri, who's going to be talking about a very interesting and difficult to study system between ulmus, a tree, and Frankia in a nitrogen-fixing root symbiosis that's definitely not the usual legume rhizobium. It's a really intractable system, and they've been able to identify some important molecules. So that's coming up next. And I hope you will follow the link that Karen will place in the chat, and that you can join our next one as well. Okay, so I'd like to turn us over to the main event now, which is thinking about how the endomembrane system, the role that it plays in plant microbe interactions, and particularly thinking about bacterial effectors. So it's a great pleasure to welcome Dr. Cécile Segonzac and Hyelim Jeon to give this seminar. And the link, I think Karen is going to place the link for the paper in the chat, because I hope that this will give you a taste. There's no way they're going to be able to do every little detail. So I hope this will give you a taste of it, and it will draw you back into the paper. And for now, I'd like you to meet the authors themselves and have them tell you about their work. Cécile.
Okay, thank you, Jane. Hi, everyone. I'm not going to say good morning or good afternoon. It's the middle of the night here in Paris. Let me share my slides. Is it working? Okay, great.
Yes, it is.
I'm going to get the pointer. Okay. So thank you, Jean, for the really kind invitation. And I also like to thank Eunsook Park for giving us the opportunity to contribute to this special issue on the endomembranes in MPMI. So Hyelim and I work in Seoul National University. And when Hyelim joined the lab, she started to look at the subcellular localization of effectors of bacteria called Ralstonia solanacerum. And she came back with this kind of pictures where the effector fluorescent protein fusion labeled what is actually the surface of the nucleus, and then those little dots, which are reminiscent of the endoplasmic reticulum and vesicles. So we got interested in learning more about this compartment and its role during bacterial infection. And we found out there's a large and growing body of studies related to this topic. So we discussed this in this review. But for today, I'm just going to try to highlight essential concepts. And then I hand the floor over to Hyelim, and she will illustrate those concepts with the few examples we cherry pick for the, let's say, time constraints. So just a few examples there. Okay, so now I should introduce what we consider plant endomembrane system. Here it goes. So it's the largest compartment in the plant cell. It connects the nucleus to the plasma membrane and to the vacuole, and it comprises most of the internal cells, internal membranes, in the cell. So we have the nuclear envelope extended into the ER, the Golgi apparatus, and then different types of vesicles that are making this link between the trans-Golgi network and the plasma membrane or the tonoplast. So we have these multivesicular bodies. They're also called late endosome. We have exocyst positive organelle, this expo here, or even the autophagosome. So there's really like a continuum of membrane here, extending from the nucleus to the extracellular medium. And the shape, the morphology or the trafficking in these membranes is regulated by their interaction with the cytoskeleton tracks. And another point is that they're also the source of extracellular vesicles, which we hear a lot about in the recent years. So these membranes, they compartmentalize a very important function in the cell. They are involved for the synthesis of lipids and proteins in the ER, and those are further processed and sorted in the Golgi apparatus. And these endomembranes are also very important for the delivery, so the secretion of those molecules to the plasma membrane and the apoplast on one side, if we would, and then also to the tonoplast and the vacuole. And there's also an important function of endomembranes in communications between plant cells. So this cytoplasmic connection here between the cells we call plasmodesmata. They are maintained by a specialized region of the ER called the desmotubules, and they're also lined by a very specialized domain of the plasma membrane. So altogether, this endomembrane system is this continuum of membranes, and so it plays a crucial role for the stress response, and in particular during pathogen infection. So these roles, they're quite obvious when we look at invasive pathogen. I'm thinking about viruses and fungi and oomycetes, which are invaders of the plant cell. So the production of the viral particles or the establishment of this feeding structure of filamentous pathogens remodel the endomembranes. So in this type of infections, we can see that vesicles and organelles are redirected to the penetration site, for example, to form this extraostoreal membrane, which has a different lipid and protein composition compared to the plasma membrane, or to secrete these callus plugs that are encasing the ostoria. So in accordance with the role of the endomembrane during this invasive pathogen infection, there are plethoras of viral proteins and fungal and oomycete effectors that have been shown to target those endomembrane processes and vesicle trafficking. And I'm gonna just refer you to a very nice annual review from the Boscott Lab, which came out very recently, highlighting how those filamentous pathogen effectors are remodeling the endomembranes. But we still are interested in bacterial pathogens, and those, most of the bacterial pathogens actually reside in the apoplast. But nonetheless, there are major cytoplasmic change that are occurring during bacterial infection. We do see change in the apoplast composition in the plasma membrane composition. We have change in the rate of synthesis and secretion processes. And those can result both from the plant defense response or bacterial activities. So first, I'd like to highlight those modification of endomembranes that are controlled by plants as a defense strategy. So the main process that comes to mind is secretion, sending weapons where they are needed in the apoplast. So we know that the perception of bacteria leads to a transcriptional reprogramming. So we have synthesis of antimicrobial compounds, enzymes, other pathogenicities related proteins that are secreted through the conventional pathways. They are sorted in the Golgi, they are delivered to the plasma membrane or the apoplast. A good example of that is the delivery of the callose synthase to the plasma membrane for this polymer deposition and the reinforcement of the cell wall. But there are also other type of secretion pathways that are involved in the antibacterial defense. There are a few reports of the accumulation of multivesicular bodies and EXPO, this exocide positive organelle, at the contact sites between the plant cell and bacteria. And both those MVBs and EXPOs, they can be the source of extracellular vesicles. Those carry proteins, lipids, and nucleic acids that can be delivered to the apoplast and maybe beyond to the pathogen here. So this role for EVs in plant-bacteria interaction still needs to be clarified, but we found interesting that the mutant plants that cannot, that are affected in the biogenesis of these multivesicular bodies are actually more susceptible to bacterial infection. So that would mean there's a positive role for those MVBs and probably EVs for the plant defense. Okay, now in parallel to secretion, the endocytosis is also contributing significantly to plant immunity. And here I'm thinking in particular about the homeostasis of the pattern recognition receptor at the plasma membrane. So those are the receptors required for the perception of bacteria in this case, and they are membrane proteins. So they are synthesized in the ER, they are further modified, they are glycosylated in the Golgi apparatus, and then they are secreted by this fusion of Golgi-derived vesicle to the plasma membrane. But interestingly, upon ligand binding, these receptors, they are internalized, this time through clathrin-mediated endocytosis, and then they transit in the early endosome or the late endosome, either to be recycled back to the membrane or eventually to be degraded. And there are interesting reports here showing that these endocytic vesicles containing the activated PRRs could actually contribute to the signaling of those receptors in the cell. Okay, and there are also other endomembrane-associated pathways that are likely involved in this plant defense response. I'm thinking about the unfolding protein response. There's a lot of there's a enhanced synthesis going on in the ER. There are also very interesting reports about autophagy, but both of them have been again reviewed recently. So I again refer you to those recent reviews if you're interested in those processes. But altogether, if we consider the roles for these endomembranes in monitoring the presence of bacteria and then executing the defense response, it looks like this compartment is really a target of choice for bacterial pathogenicity and virulence. And this is something that can now be visualized. So tools were developed to monitor the networks of endomembrane during bacterial infection. For example, here we have this transgenic Arabidopsis line that is constitutively expressing fluorescent protein retained in the ER through this HDEL motif. So in this line, you can see the classic morphology of the ER. We have this really large network of membranes that are forming either tubules or more flattened sacs that are called cisternae. And with super resolution microscopy, we can also distinguish different ER domains also at the contact sites between those membranes and the other organelles in the cell. And interestingly, the morphology of the ER is profoundly changed when these plants are challenged with bacteria. In this case, it's Pseudomonas syringae here that is labeled in green. We can see a few microcolonies here in contact with the plant cell. And within a few hours after infection, these authors are reporting collapse of the tubular networks of the ER. And I found that was a very interesting study because this is not seen in the cells that are not in contact with the bacteria. It's also not seen in cells that are treated with purified flagellum. So that's a good indication that this modification of the ER morphology or the ER structure is an active process driven by the bacteria, maybe in the aim to inhibit or redirect the secretion pathway. And we have another interesting study that kind of correlate with this one. This time about the formation of ER bodies during Pseudomonas infection. So these ER bodies, you can see them here. They are this kind of, as a specialized region of the ER. They are forming these elongated blobs on the tubular structure of the ER. And these ER bodies, they are formed through the action of a specific shaper protein. So those proteins that can modify the curvature of the ER membranes. And it's also positively regulated by jasmonate. So these ER bodies, they were studied in other conditions. They are actually they contain beta-glucosidase enzyme. They can release or contribute to the release of glucosinolate during the plant-insect interaction. So they contribute to anti-herbivory response in brassicacea. Now, in this new study, what we see here is that the ER body formations, all those little green dots here, is also induced by bacterial infection. So when the plant cells are in contact with bacteria, in this case, bacteria that can produce a J mimic, the coronatin toxin. And these authors also found that the plants that were impaired in the ER body formation, so the mutant of this shaper protein, for example, displayed enhanced bacterial resistance. So that's a very interesting phenotype, which suggests that these ER bodies may act as a susceptibility factor for the plant, or at least that they contribute to the virulence activity of this coronatin in the brassicacea. And there's a last way we thought bacteria could interfere with endomembranes, and this is through the cytoskeleton. So these cellular tracks that are guiding the membrane networks, especially for the ER. So again, we can monitor the morphology of this cytoskeleton network during infection by using actin binding protein or microtubule binding protein domain fused to a fluorescent reporter. And here, for example, the researchers observe the bundling of actin filaments in response to the virulent Pseudomonas syringae strain. And this modification of the actin filaments was dependent on the type 3 secretion system. And similar studies have shown that the microtubules also are disorganized during infection. And these studies, they are backed up by pharmacological approach where the researcher experimentally disrupt this cytoskeleton network, in this case by using chemical here. This oesaline is preventing the microtubules to polymerize, and you can see that plant treated with oesaline are more susceptible to bacterial infection. And this is again depending on the type 3 secretion system. So these all those studies, they suggest that there are bacterial factors that actively disrupt these cytoskeleton networks to promote infection. And we wonder if this modification of the cytoskeleton can also impact or actually aim at impacting the endomembrane function. So if we consider this central role of endomembranes for the plant defense, and then all those modifications that are driven by bacterial infection, there was plenty of ground for Hyelim and me to look for bacterial effectors that target endomembrane in the plant cell. So we compiled a list of bacterial effectors that localize to or associate with endomembranes, mostly using two different large-scale studies that were recently published. So both those studies are investigated the subcellular localization of effectors and the effectors targets with data that were obtained from live imaging or different types of biochemical approaches testing protein-protein interactions. So the meta-study from the Devo and Gutman lab in Toronto could highlight that proteins that are interacting with type 3 with the bacterial effectors are enriched for geoterms that are related with endomembranes, with the plasma membrane mostly, but also with ER, vesicles, and the cytoskeleton. And on the other hand, we have an interactome study from the Peters and Noel's lab in Toulouse, where this study revealed that the certain hub proteins, so there's proteins that can interact with several different bacterial effectors, are actually associated with endomembrane. So that's the case here for this Exo70E2, and that's a component of the exosys complex. This is a protein complex that contribute to the fusion of vesicle with the plasma membrane. And it's interesting to see that this protein can interact with several effectors from Ralstonia solanacearum in this case. So in complement to using these two studies, Hyelim and I also skimmed the literature. We were looking for evidence that these bacterial effectors contribute to virulence. So to make sure this association of the effectors with the endomembranes is actually relevant for their biological function. So at the end, we came up with a shorter list that you see here. And next, we tried to categorize these effectors depending on their mode of action, or what is known of their mode of action, trying to understand how the host endomembranes can be manipulated to the benefit of bacterial pathogens. Some of these effectors are acting on the secretory pathway, some are acting on the cytoskeleton, and some don't. That was quite interesting. And now I'm gonna let Hyelim give you a few examples that we selected from this table. And I'll come back for the conclusion. Hyelim, I hand you the floor.
Okay, thank you, Cécile. And yeah. Okay, so I'm going to show you a few examples from our reviews. I cannot go through every detail, but you can find it in our reviews, or please leave the questions here. So let me start with the effectors that block secretion. As you saw in the table, inhibition of protein secretion or callus deposition is the most noticeable function of effectors that localizes to the endomembrane system. These two assays, easily detected by fluorescence microscope, are used for screening effectors with these virulence functions. For the protein secretion assay, fluorescence protein fused with a secretion signal peptide is used as the reporter. It goes through the conventional secretory pathway from the ER to the apoplast, where the fluorescent signal disappears. These three effectors, HOPE1, ZOP-B, and ZOP-J, were found to suppress secretion of this reporter, resulting in stronger signals stuck in the ER. And these effectors also inhibit callose deposition induced by the flagellin peptide, FLAG22, or Bacterial. Interestingly, these three effectors seem to have different virulence mechanisms to end up with these same phenotypes. For example, ZOP-B localizes to the Golgi network with its targets or functions they are unknown. And ZOP-J is located on Golgi and the plasma membrane, and it inhibits the proteasome function to suppress immune signaling. And HOPE1 localized to the microtubules, and there it interacts with its target proteins, which I'm going to show you in the next slide. So these observations imply the link between secretory pathway and cell-based defense responses. So impaired secretion may block delivery of callosynthesis or other defense-related molecules, resulting in impaired defense. So this is HOPE1, and in case of HOPE1, it seems that inhibition of secretion is directly related to its virulence function. As mentioned, HOPE1 localized to the microtubule network and interacts with this microtubule-associated protein MAP65-1, which is important for microtubule bundling. So through this interaction, HOPE1 dissociates MAP65 from the microtubules, leading to the defect in protein secretion. MAP65 also found to function in immunity, as MAP65 mutant display reduced secretion of antimicrobial protein PR1 and enhanced susceptibility to pathogens. These are great evidences for effectors disturbing cytoskeleton regulators such as MAPs and actin-binding proteins to directly inhibit secretory pathway and extracellular immunity. And next one is HOPE-C1A. It has the similar yet different mechanisms to interfere with the microtubule networks. It localizes to the plasma membrane, where it binds and acetylates tubulin, which is the major component of the microtubules. So this results in dramatic disruption of the microtubule network and blocks secretion and callose deposition. And HOPE-W1 is the first bacterial effector found to directly target actin, another main cytoskeleton. The incubation of actin filaments with HOPE-W1 increases the amount of soluble actin in the supernatant and decreases actin in the pellet, meaning the depolymerization of this filamentous actin. So it results in disruption of actin cytoskeleton and actin-dependent vesicle trafficking, such as endocytosis. So these two effectors support the important role of the cytoskeleton in different vesicle trafficking pathways during plant immunity. And there are other effectors that can modify vesicle trafficking, not disturbing cytoskeleton. HOPE-M1 is the first effector found to localize to trans-Golgi network, which has been studied very well until recently. This effector has multiple target proteins called HOPE-M1 interacting proteins, means in short. And among them, means7 is one of the regulator for GTPases that are essential for overall vesicle trafficking pathways. So HOP-M1 degrades Min7 through host proteasome, resulting in immune suppression, including callus deposition. So what is interesting is that the presence of HOP-M1 or deletion of Min7 can induce an aqueous apoplast shown by these water-soaking lesions, which is also crucial for the virulence. Taking all these observations together, we can correlate the manipulation of vesicle trafficking with the aqueous apoplast during plant infection. Although we don't know yet which cargo proteins are affected by HOP-M1 and Min7 interaction, but it leads to higher water content in the apoplast, which would provide better environment for the bacteria, probably by diluting antimicrobial compounds such as callus, or it can facilitate nutrient flow to the bacteria. So further characterization is needed for the clear explanation, but HOP-M1 has become a great cellular probe to provide us new insights about plant immunity and bacterial infection. And finally, I like to show you two effectors that function independently of secretion or cytoskeleton disturb. HOP-D1 is the ER-resident effector that interact with the membrane-tethered NAC transcription factor NTR9. These transcription factors are tethered at the ER or plasma membrane, and it is released from the membrane by proteolysis in response to multiple abiotic or biotic stresses. The cleaved NTLs are imported into the nucleus to regulate the expression of stress or defense-related genes. NTR9 was shown to positively regulate plant immunity, inducing expression of multiple defense-related genes. Interestingly, HOP-D1 suppresses expression of these NTR9 regulated genes only when induced by the avirulence effector AVR-RPM1. But it has no effect when these genes were induced by FLAG22. So how HOP-D1 modulates this NTR9-dependent transcriptional regulation is not characterized yet. But this implies that endomembrane system also participates in a feedback loop of stress responses from sensing stresses, signaling, and to regulation of transcription and back to translation in the ER. So HOP-D1 may interrupt somewhere in between this loop to specifically suppress effector-triggered immunity. Besides the intercellular trafficking, cell-to-cell communication through plasmodesmata also plays important roles in plant pathogen interaction. These studies have come up only in the past few years, starting with HOP-01-1 for bacterial pathogen. This effector was found to localize to the plasma membrane and plasmodesmata marked by this callus staining. Here it interacts with plasmodesmata localized proteins that are important regulators of callus homeostasis. Through this interaction, HOP-01 destabilizes these proteins in a proteasome-dependent manner. Interesting thing is that in HOP-01 overexpressing plants, diffusion of YFP protein from one transformed cell to neighboring cells is promoted. In addition, HOP1 deletion mutant could not spread well from the infectious site to the distal tissues compared to the wild-type pathogen. So this suggests that HOP1 manipulates plasmodesmata-mediated cell-to-cell trafficking by destabilizing callus regulators. So then how targeting the PDLPs can promote infection? This is the basic composition of plasmodesmata. Around the plasmodesmata neck, the plasma membrane is enriched with these plasmodesmata proteins, including PDLPs. So they modulate callus deposition here, and it results in alteration of PD aperture. This mechanism is not revealed yet, but overexpression of PDLPs causes reduction of intercellular movement of this mobile effector HOP-AF1. So these findings suggest that PDLP positively regulate PD closure to inhibit effector movement, and this process is targeted by HOP-01. And besides HOP-AF1, there are 15 more DC3000 effectors shown to move between plant cells. So identifying the function of these mobile effectors in plant immunity will help us understand how pathogens colonize and spread from cell to cell. So this is that is all for me, and I'm going to go back to Cécile for the big summary.
No, don't stop sharing. Can you put back the slide?
Yeah, I forgot.
I just wanted to conclude with the next slide. Can you put it up? Yeah, thanks. So we've seen like there are many different ways that are emerging for this from those functional characterization of the bacterial effectors, how they can affect plant immunity, but also other process in the cells, maybe to their benefit. So Hyelim made this really large figure for the review. We refer you there for more details. I just wanted to add up that since there are first a few important cases we didn't show today, but there are growing evidence that the exocyst complex is an important target of effectors, especially bacterial effectors. And there are a few more studies that came up after our review was published that also highlight effectors from other bacteria than Pseudomonas. We've seen a lot of Pseudomonas effectors today, but effectors from Ralstonia, from Xanthomonas, from Acidovorax in this AOW1 case are also shown to target vesicle-associated protein or to modify the ER and the cytoskeleton morphology. So we think that's going to be a very important area of research now and in the coming years. And then, can you put the last slide, Hyelim? Just to finish, when we were putting together this review, we thought there was a few points emerging from from this work that we'd like to share with you. A first one would be kind of mechanical, but we are interested to understand how the type 3 secreted effector can associate with membranes inside the plant cell. It's been shown for a few of them, they can be acylated, but many of the ones we are looking at in my lab do not have acylation site predicted, so they might be interacting with the membranes through interaction with these endomembrane components or proteins. Another interesting point is that these effectors, they can now be used as probes. So I'm thinking about the effectors like OpW1, for example, that interact with the cytoskeleton and modify the morphology of the ER. Those are very interesting tools we could use to gain some fundamental knowledge about this relationship between the cytoskeleton and the endomembranes in the plant cell. Or as Hyelim mentioned with the OpM1 story, we can also use those type 3 secreted effector to purify the vesicles that they are labeling to figure out their content and their role for plant immunity or for the promotion of bacterial growth. And then there was one more question that we were particularly interested in with Hyelim, is this selective inhibition of the secretory pathway. What I mean here is that obviously it makes sense for pathogen effectors to suppress antimicrobial secretion to the apoplast. But at the same time, there are also membrane proteins that are required for bacterial promotion. I'm thinking about the aquaporins, for example, or maybe the sugar transporter that need to be delivered at the plasma membrane to help for the release of nutrients in the apoplast. So we are wondering if by characterizing more bacterial effectors, we can find out how these vesicles are sorted, how they are marked. There are specific signals that would decorate vesicles carrying membrane transporter, for example, or the vesicles carrying the antimicrobial compounds or the PR proteins. And the very last point is that we really focus on this secretory pathway in the review, but it's been known for a very long time in animal pathosystem that there are other ER processes that are targeted by the bacterial effectors, especially this unfolded protein response or the ER stress pathways. So I guess it's just a matter of time before we also identify effectors from plant pathogens that also target these processes. Okay, so I'm gonna leave it here to this with this open question, and thank you all for listening to us today. Thank you very much.
Thank you, Cécile and Hyelim, for a really excellent and interesting overview of this big new field. I particularly love the idea that you raise about using these type 3 effectors as a way to learn more about these connections between the ER and the cytoskeleton. It reminds me a lot of the way we've learned so much from viruses about plasmodesmata and how they work. and that these tools, we end up, as we try to study viruses, we end up, in fact, learning an awful lot about how plant cells work. And I think that you're exactly right that these type 3 effectors are going to be a new set of tools for understanding the complexities of the trafficking system and connections with the cytoskeleton. So really, really interesting and a lovely way to synthesize and pull this all together. So thank you very much. Let's take a look at the questions. So I see people are starting to type them in. Please add your questions directly to the Q&A box, and I will read through them one at a time. So Jesse Kaleku asks, is there any reported case of these phenomena induced by fungi? So targeting, I think, of the secretory pathway.
You want to go for it, Hyelim? No? Right. So yes, actually, that's also field that's been touched on by the filamentous pathogen community in the recent years. I was highlighting this review from Tolga's lab because in the case of fungal pathogen and omicid pathogen, there's obvious need for this endomembrane remodeling. There's new membranes that are being formed. There's a actual deformation of the plant cell that we don't see in with the bacterial infection. So there are also large-scale studies with omicid effectors targets that also seem to have hubs in this as endomembranes components. I think we do cite some of those reviews and studies in the introduction of our review, because we use this point to say, okay, we know it's actually occurring when the plant cell is invaded by this feeding structure or the hyphae of fungus. And it's not that obvious why the endomembrane would be remodeled during bacterial infection, because those are just thriving in the apoplast. But obviously, that's not that simple.
Yeah, no, that's a good point, that the fungi are invading much more, although there are intracellular microbe bacteria sometimes.
And yeah, we put aside the molecules. That's very true. I'm sorry for people interested in that.
But it is true that delivering so many things, like the whole idea about those sugar transporters, the sweet transporters, delivering those, or the aquaporins to the surface is so important. Okay, next question. Deepak asks, are there effectors known which target secretory pathways to manipulate cell wall sugars, which directly follows from this conversation we're having?
Can you repeat this? Sorry.
Are there bacterial effectors which target secretory pathways to manipulate cell wall sugars? So I think, I'm not sure whether they're asking about the composition of the cell wall. I guess there's callose, but I was thinking more about delivery of sugars into the apoplast. So I think you could think about either question, really.
Oh, it's a really good point. I guess it's a matter of techniques now to follow, for example, the sweet transporters, or the ones that we know are accumulating in response to virulent xanthomonas, for example, to follow them with the live imaging of FRET, see if they can be delivered properly during infection with this time Xanthomonas strain that would be deleted for this effector or expressing effectors known to interfere with vesicle trafficking. I don't think we came up across this kind of study while we were preparing, but that's definitely something that can be investigated, and this is something we're interested in. I'm just going to make one point about Hyelim's work quickly, but she found a factor that could seem to affect the PR1 secretion, but not this SEC-GFP reporter secretion. So we were a bit puzzled how maybe certain vesicles can be targeted and not others, and how to distinguish them when you're a bacterial effector.
That comes back to that earlier point that I think that these effectors are going to be really great tools for perhaps helping us to identify different subsets of vesicles and vesicles pathways. So it's a really good question. And I think you're also pointing out areas that of research, right, that people could follow up on. Okay, Pawan Kumar asks, are these effectors released at, this is all about timing. Are the effectors released at a particular time point during infection, or are they secreted from the start of infection to alter endomembrane system of plants?
That's also a very interesting question. First, in general, because we're still, I mean, beside Pseudomonas and even for Pseudomonas syringae, I don't think there's a lot of deeper study about the delivery, the timing of the type 3 secreted effectors. There are a few, but we need more, especially for other bacteria. For Ralstonia or Xanthomonas, we really rely on transcriptomic or of the expression, of these effectors during infection. To answer this question, I would just say that the modification of the ER or the cytoskeleton that are seen during infection with the virulent bacteria and not with the type 3 secretion mutants, for some of them, they are quite early. So the ER study, I think it's within 6 or 7 hours, the authors start to see the collapse of these tubules in the ER. But now to think about the order of delivery for the bacterial effectors, I think there's a lot more work to do. So they may act very early. They would like probably to, I mean, one of their aim is likely to prevent the secretion of the antimicrobials, but this process takes also several hours for the plant to reprogram and start producing those molecules. So there's very interesting kinetics there to study, get more insight in this battleground.
Yes, and certainly we know there are ways of like things that are produced early, mid, later. So it makes sense that it's like a carefully orchestrated set of interactions where the plant is first the bacteria is trying to change something this way and then that. And I think also we have to think about what happens with the levels of these effectors. What happens when you have a small amount? How does that affect these endomembrane systems? And does that change things on the surface? So I think, yeah, I think as we start looking at this in a much more time-dependent way, it's gonna uncover some other really interesting things. I think, so just to remind you that if you have, if you're typing things into the chat, to please type them into the Q&A so that we can, and I think we have another one, so that we can answer them. But I do see one that's still in the chat, so I'll just read that off there. Timming, who asks, are there any effectors that could lead to the collapse of the ER? And I think that circles back to some of the work you were talking about at the beginning. Maybe you could recap that for a moment.
So I actually cite the bioRxiv version of this work. It's now published, but the author use the fluorescent Pseudomonas to show that the ER morphology changed during bacterial infection. But then they switch to another system, and they look at I think it's fungal or oomycetes effectors that can target the ER directly. And they identify actually a very interesting motif and fold of these effectors that enable them to interact with the ER membrane and probably lead to this remodeling of the ER. So in the case of the bacterial effectors, I don't think of anything from the top of my head. We know that the HOP-W1 in Pseudomonas, or its homologue in Acidovorax, is actually both interacting with the actin cytoskeleton and also associated with the ER. So I don't have a name for an effectors that could lead to the ER collapse, but there are definitely a few that have been identified, and they're very interesting to follow on to see this relationship. And also, if I'm thinking those effectors, they could interfere with inhibit or block the action of this ER shaper protein. So this is another interesting place to look for a way to actually collapse the ER, or maybe what we interpret as a collapse is actually a redirection of the whole process that are taking place in these membranes.
It's a good point. Oh, we have two more. Deepak asks, with respect to the arms race between plants and pathogen, are there clear examples of changes or adaptation of the secretory pathway to counter pathogen effectors? In other words, have we yet to identify a GARD or GARD for various endomembranes? I'm not sure we're there yet.
I think there's one example, right? Hyelim, do you think about this TN2? So it's not a full story. So, but there are clues that so TN2 is a TR and B protein, so it's not a full NLR protein, but it is associated with the ER membrane, if I'm correct. And it seems to guard the exocyst complex, or some of the subunit of the exocyst complex. So mutations in those subunits can lead to dwarfism or cell death in plants, and this is through the action of this TN2 that localize at the endomembrane. But that's, I think, the only example we came across. For the plasma membrane, there's like a lot of examples, but for the ER, I I don't know if we have other NLR there that can be guarding endomembranes component, but that would be a good place to be, I guess, also for those receptors.
Interesting. Yeah. Makes me think also of rhizobium legume symbiosis, where you have that exos 70 something I or something like that, which is required for the infection thread for the actual entry, the beginning of that infection structure where the bacteria will enter. And there's definitely an association with the nucleus and yeah, all those inner parts of the cell connected and interacting in ways that previously we could only just see like on an EM or something and the microscopy. And now we have these proteins that we know localize to these spots. Really exciting. Okay, Tuan Tran says, very intriguing talk, Cécile and Hyelim. Is there anything known about how HOP01 can localize to plasmodesmata and interact with PDLP5?
That's one for Hyelim. Do you know how HOP01 localized to the membrane and accumulates in this plasmodesmata?
I can recall, but about the interaction, the ADP ribosylation may have some role in degradation of PDLP5 and 7, but the exact mechanism is not known. But ribosylation site of PDLP is important for their degradation.
No, that's interesting about that ADP ribosylation. And Iris Molhoff asks, are there effectors that impact the formation or disruption of lipid rafts or plasma membrane microdomains? Oh, that's a really interesting question.
It is, and it would probably explain also how some of these effectors can localize to very specialized region for the plasmodesmata, for example. But when you survey the bacterial effector sequences, there are really not many that have transmembrane domains or transmembrane region or this acylation residue that could be acylated to directly bind to how do you say, insert in the phospholipid bilayer. It's likely that this recruitment to the membrane is through protein-protein interactions. There can be just electrostatic interactions or actual interface between those proteins that we know are enriched in the nanodomains, for example, or this specialized plasma membrane in at the plasmodesmata. This is not your usual plasma membrane like around the rest of the cell. So these nanodomains, they are enriched in specific lipids, but also in specific proteins that could be the anchor or the target for those effectors to be recruited to those regions. And that might be what we see with OPM1 that targets those MIN7 decorated vesicles. There's actually a lot of other OPM1 interacting proteins that are localized in the same complex. So some of them might be the actual target of the effector. Some of them may help to reach particular compartment for those effectors.
Yeah, it's a really
There's a
good
lot more to do, but
question.
I think now we have a lot of really great tool with the super resolution microscopy or these purification methods for vesicles. We have everything in place.
Yes.
Let's go.
So what do you think? We still do have other questions, but I think given it's we're past the hour, what do you think? Let's end with a question of what do you think are the next sort of areas that people are going to be probing or that we should be thinking about as we look forward to the future of this field?
It's a tough one, Jean, but I work a lot with bacterial effectors, but I actually come from, I studied cell biology in university, and I'm in the plant side. I'm really interested in how we can dig into those vesicles, how we can separate them, how we can get into their content, and how they are sorting out those molecules with different fate in the cell. So I really see the effectors as probes. They can help me grab the vesicles I'm interested in. So there's a lot of fundamental knowledge that can come from this.
Yeah, I think that's actually, to me too, that's one of the most exciting things that you brought up, this idea that we can learn more about the whole vesicle trafficking, targeting, and what are these different compartments, that they may distinguish things, allow us to get another layer of resolution than what we've had before. So very exciting. So I think we'll stop there, although we do have 2 more questions. I just want to remind people that when you go to the article in the journal, that there's one of the authors is a corresponding author, and their email is there, and you can always reach out to them with your questions. So let's thank Dr. Cécile Segonzac and Hyelim Jeon for an excellent talk and a very interesting and valuable resource in this review article. And I see some clapping coming up virtually. And I hope that you will take a moment to go and look at this review article. You saw some glimpses of the very helpful diagram and figure that they created. And thank them for joining us for What's New in MPMI. So we'll stop there. Lots of positive feeling here. Great questions from the audience. And remember, you'll be able to check back probably tomorrow and see the recording posted. And I hope that you will share that with colleagues who were not able to watch it live. Okay, thanks everyone.